Replenish yourselves with food and some coffee. So this is the last, we're coming to the end of the wonderful workshop, but we have some very exciting titles coming up. So for this afternoon session, there will be two speakers. The first is Arik Yochelis. I'm not pronouncing it correctly, but he's from the Ben-Gurion University, and he'll be talking about pattern formation by molecular motors and cellular protrusions. And I saw some very exciting slides there, so very much looking forward to your talk, Ben.
Thank you, Veronica. Thank you guys for staying after lunch to the last moment, right on. Since already some of you expressed confusion about the landscape and the Swiss Institute, so let me explain a little bit before we go to science. This is an arid area of Israel, south of Israel, okay? So it's not the Swiss borders. We have been donated a lot of money for the institute from the Swiss government, and therefore we are also Swiss Institute, but it's not Switzerland. Okay. There's nothing that someone put in your food, it's okay. Just to clarify that, so this is half of Israel. Tel Aviv is around here, Jerusalem is here, Beersheba, where the main campus is here, and we have another campus in Sde Boker, which is devoted to desertification by any means of that it sounds. It has to be with the plants, the demographic issues, energy conservation, energy generation, and so on and so forth. It combines a lot of people, ranging again from sociological, anthropological disciplines, and to applied mathematics and physics. It's a very small campus with a very small village, so it's really kind of small paradise or oasis in the middle of nowhere. It is designed really to adapt to the desert. We have quite an unusual equipment. This is the largest solar concentrator in the world, by the way, so this is the scale, this person here. And we have also very unusual faculty, which is also good. There are a lot of things to do. I mean, it's not only a dry desert. We have a lot of water, a lot of beautiful landscapes. We have floods in the winter, which is a remarkable phenomena. If you haven't seen a flood in the desert, this is really something remarkable. You see the waterfront really propagating to you. It's like if you saw Lord of the Rings, it's really something like that. And we even have tulips. And sometimes in the winter we have all these regions are red because of the bloom of the tulips. So if you have the chance to visit, I urge you to do so. Okay, so let's go now to science. So why cellular protrusions? So, we have heard here a lot of talks about cells, and there were also implications about protrusions, or like the philopodia, for example, even today. But protrusions, cellular protrusions, are much widely more happen in our body. This is another example, before I go to philopodia, of something that is called stereocilia, and this is a vital organ to our auditory system. And the mechanics of this stereocilia is very important to detection of sound, and they play a cardinal role in how we hear. And of course, the mechanics of it is very vital, and I'll show you it's being dictated by very small creatures that we will refer to them later on as molecular motors. The other example, and again there are many, but I'll focus only on this one, is the philopodia. Philopodia is all these strange antennas that go from the cell. Has been studied widely by different methods and disciplines. This is just a cartoon, of course, but it's highly important to communication, to migration, to wound healing, and so on and so forth. Okay. Now, everything that is interesting here is exactly these kind of structures, because they are infrastructure or roads to something else. And something else is the molecular motors that I will be referred to very soon. The size of these creatures, of the Philopodia, and also the Stereocilia pretty much, is microns or tens of microns in length, and usually tens of microns in diameter. And again, this is a rough estimate. It can be, there can be deviations as well. So, my outline will be as following. I'll first introduce you to actin-based cellular protrusions, exactly as I showed you before, which are the Philopodia. I'll introduce you again to molecular motors, basically myosins, and I'll show you the role in exactly what they do inside of these cellular protrusions. I will not go too much into the cell itself. I will focus only on the Philopodia itself. I will show you that they really also have the pattern formation properties by forming aggregates. This is quite interesting and unusual, and this will be, in fact, the main talk and the analysis that will follow up. So the next step will be to introduce the theoretical framework, and I will introduce a model, but it's not really a model for molecular models that I'm interested in. I do want to establish a theoretical framework that you can take later on and to transfer it to other problems as well. So the model will be rather primitive. It will capture, as I will show you, the most essential properties, but it will not be restricted to only molecular motors. I'll show you the analysis. I'll compare with the experiments, and at the end I will show you the outlook that will kind of envision a little bit more beyond what is shown here, and of course I'll summarize. Okay, so you can construct different strategies how to represent Philopodia, and what I want to focus on is in principle this network of actin filaments, which, if you think of it as the boundary of the cell, so it can be quite randomly distributed in the cell itself, but then when it grows these protrusions, it becomes very ordered and elongated in the same direction. And it happens mostly because there is a polymerization process, and the alignment is a rather spontaneous behavior. Again, it needs to be triggered, of course, but again, I'm not, only at the end I'll say a little bit about what happens and how it aligns, but right now I really want to focus what happens here. So here, what happens is that we have this kind of strange molecular structures, which very often referred to as motors, and these guys, which are also known as myosins in a biological context, they just walk on top of these filaments, and some of them, well, not this, but they use their energy to contract because they are pulling the filaments and they contract the cells, while others, what they do is they deliver chemical cargo from inside to the exterior of the cell. So this is the mean of the transportation of the information of the cell from inside, outside, and then again inside. Now, you may ask, well, why do I care about such small objects? I mean, we all saw that several talks about Turing and mean fields approach and so on and so forth, and I'm really a person that comes from mean field approach and pattern formation, and yes, we do need to care about these guys. And I will show you that mean field approach is really also valid for these small creatures. So we can have multi-scale approach even if we talk about molecular objects. So here's one experiment which is, I find, particularly interesting because it's very difficult to do in the sense of the lab. So these guys here, these are not us, of course, they put a quantum dots on top of these motors, and they were able to illuminate and photograph traces of motion of these individual objects. Okay, so we're talking about molecules, or several molecules that being photographed and traced later on. So these motors can have the ability to go back and forth, and very often their motion is related to what cargo or what chemical entities or molecules they carry on them when they travel. Here's another picture from a rather famous Nature paper, and it shows something different. And I'll try to convince you that it shows something different. So what you see here, let's say that we don't care what really happens here. What we really care is that this is the end of the cell. These are the philopodia. It's very vaguely shown here, but this is the end of one of them, this is the end of the other, and so on and so forth. Here it's shown a little bit better, and what you see here is that these kind of dots, and I will show you such structures again, and they're all propagating. For example, let's focus on this one. It's already here. So the philopodia itself remains the same. It keeps its length, but we have propagation of these dots along the philopodia structure. Now, you can think of it as a single molecule, but it's not. And here's another figure which surprisingly appears in the supplement of this paper, which I find it maybe the most interesting figure in this paper, but again, I'm not a biologist. And it shows very clearly that you have traces of low density, which correspond to small density of molecules, and you have here traces of large densities. And these are kind of resemble to traveling waves, right? And they all go from the tip to the cell. So this is time here, and this is the length of the philopodia. So they're going from the exterior to the interior. Okay? So we have already an individual motion and a cooperative motion, right? But things are a little bit more complicated, because sometimes you can really observe a traveling wave type phenomena. So again, this is the end of the philopodia, this is the cell here, and these guys are travel from the exterior to the interior. And sometimes you can have only one. So it's not really a robust behavior, okay? So sometimes traveling waves, sometimes isolated pulses. Now, if you think about it from the dynamical point of view, these are really different qualitative, different origins of different behaviors. You can think a bit of that one as oscillatory motion, and this is like excitable motion, like what happens in our neurons or in our cardiac system, the action potential. And I'll show you that it's actually, you can capture everything in the same framework. So the basic idea in biology, at least to me, is to really identify the correct questions. I mean, biology, as many of you guys already mentioned, is absolutely enormously complicated. And it's really, it depends how you tackle the problem and what kind of questions you aim, that you will be able to provide reasonable or unreasonable questions. And we very often face by biologists that will take a mathematical work and will say, okay, that's just mathematical crap. I mean, that makes no sense at all. And very often, mathematicians do say something useful. I mean, Alan Turing was not a biologist, and say something very fundamental that how many talks we heard here, and it's not only because we are all inclined to mathematics. You can see the same phenomena also in biology. Hodgkin-Huxley, it's a computational work. It made a revolution in neuroscience, and one of them was a physicist. So very often it is useful to take or collaborate with a mathematician or physicist to make a difference. But very often, many of our works, let's acknowledge that, is written and being forgotten almost forever. Even though that we claim that we have resemblance to some solution to biological system. So the basic idea, and Veronica referred to this slide when she said about attractiveness. I'll try to convince you that it is a symmetry breaking, very similar what happens in the traffic jams that also happens in Philopodia. If you ignore the circular structure and you open it up and put a periodic boundary condition, so you just have a 1D system, at some point, what happened here, you have a symmetry breaking and you will have a traffic jam that is being produced, and it can only propagate, but it will persist forever in this setup. So you see that cars are going this way, the traffic jam travels the other way around. If you think about each individual as a car, then in principle we set up the stage for understanding these bright spots of molecular motors. So what are the key questions that I want to focus on? So the first of all, I want to understand what will be the most reasonable phenological or physical framework that I should adopt. That's the first one. Should I have local, global interactions, autocatalysis, Michaelis-Menten, and so on and so forth? The other one, I do want to have some notion about, is there a biological advantage in the mechanism that I will provide? I mean, if I can only reproduce these bright spots, it's good, but it's not enough. I need to provide some biological insight that to allow this model to be a little bit more vital. And of course, robustness and how generic these insights are. I mean, can we apply it also to other systems, and is it really sensitive to model details? And hopefully that I'll convince you that I can provide insights to all these questions. So now we move to the second part, and this is the modeling part of the theoretical framework. So the basic idea is to understand whether we need self-organization or emergent phenomena, or the molecular processes where we count by molecular dynamics or some other means the phenomena itself. So in principle, what I will try to do is to be here, not here. Then I end up with pattern formation theory, and you all know these pictures. And the nice about these pictures is not that because they are colorful and beautiful, but because you can have universality and self-organization explanations for almost each one of them, even if you don't know the mechanism itself. Now, self-organization is actually a very old problem, and I was surprised when someone gave me this citation. This is, by the way, the Hebrew wording. This is the English translation, and it was already organized in biblical times, right? But the fascinating thing is that it really provides universality. So for example, you can have stripes or morphology of stripes and defects at pigmentation of animals, sand, skies, and if I omit the scales, you won't be able to distinguish between the patterns. And this is what I did here. So this is a chemical reaction. This is a slime mold dicotyledon. These are intracellular calcium waves, and I on purpose rescaled them, and you see that if I would omit the labels, you wouldn't know which one is which. So the fact that we can use universality, it's a very strong power to understand qualitatively the mechanism that we are maybe dealing with a certain system. There's too much light, so hope that you will be able to see the borders of the movie will be here and here. So what you see here is the membrane of the cell is here, and all these guys are the filopodia. And I'll try to run it a few times that you will see much better. So that was done by NIH colleagues of us, and I use a lot of time to analyze this data to understand and extract from it useful data. So you see all these are the ends, and you see here a lot of behavior that is going on. So without too much ado, here's after many hours of spending and looking at these movies, here's what I thought that I found. So we're looking at a specific filopodia, and all these traces are densities of these motors. At first, it looks like it's quite random, okay? But if you look a little bit more carefully, you see here few features. One is that you have a very sharp peaks that are being initiated from the tip and then travel to the base of the filopodia. This is one scenario. If you track them, it's shown really beautifully, and I'll show you space-time plots later on. The other one is that you have a very thick region that breaks up, and you see it here, and then triggers waves. Okay, so we potentially you have here two behaviors, and you can track these along different times and the different filopodias. So here's another event of breakup that you see here. But again, this is not a very conclusive. I mean, you can conjecture a lot of things that it's due to noise and many other artifacts. This is not conclusive, which is true. That will be a very fair conjecture. This is exactly what I will want to try and mathematically show that this is not an artifact. This is a true phenomenon. This is for a shorter filopodia, another example. So this is just initiation of a pulse, and this is a breakup into wave of a wide region. Okay, so let's go to model and analysis right now. So I will try to express a very simple and maybe naive model, but I will show that it captures the qualitatively valuable information that we can proceed with that later on. So what we do is the following. So the biological feature of the filopodia is that it has polymerization of actin filaments from the tip to the base of the cell. On top of that, there are the myosins that are like in the subway staircase. They just sit here, and therefore they are moving with the propagation velocity of the actin filaments, right? There is another type of mode of myosins at which they propagate a little bit faster, and we all, as grown-ups or as a children, try this trick to climb this staircase faster than it moves downstairs. And there's another mode which is a little bit more negligible and happens at low probability, but these guys can also diffuse. So they just detach from the actin filament, and then they can just randomly diffuse. So we have three species. Again, a very generic one. One mode that is going towards the cell, the other one that is going towards the tip, and one that is freely diffusing, and we assume that we have transition rates among each one of them. So a very simple chemical reaction. If we look at the equations, then we write equation of motion for each one of them, and we designate the functions here. So these are the fluxes, and these are the functions. It's very similar if you think about it, and this is what I like about this approach, is that you can find similar approaches in other fields. If you look at micelles, which are just lyotropic structures, they can diffuse freely as uncharged species. They can interact and become charged. I'm sorry. And if you apply electrical field, they will go to the other side because of the electrical field and interaction with it. So it's basically exactly the same picture. So I don't really need specific details to explain or to describe it. Okay, so but I do want to give you a little bit more details. So the fluxes, for example, for the freely diffusing will be just a simple diffusion. Here we assume just an advection. Here also we assume an advection. All the other terms are just linear crossing from one mode to the other. And what we really want to assume is that something about this guy, which is related to the actin filaments, and this guy is really has the interaction of cooperation. What I wanted to say is the following, that if I'm going in this direction, then I can move within the small density of people and cross them and then move freely. This is these guys here. But if I'm stuck with a lot of people here, then I will move together with them even if I want to go downstairs. So this is the basic physical principle, and this is the lowest term that gives you this individual conversion that you want to go with the main team and not against it. Okay? And you can take other polynomials and it will not qualitatively matter what exactly you do. And I will show you mathematically why is that. Okay, so again, let's compare it to other features. So we have this kind of models almost everywhere. We have it in fluid dynamics, in chemical reactions. We have it in biology like somatogenesis, in ecology, in the problems of vegetation because of a runaway of water. We have electrokinetics like electric ink, and so on and so forth. So this is really a generic framework. And this is referred to as reaction diffusion advection. It's not reaction diffusion, but only because it has an advective field, it is reactive diffusion advection. Now it has very two subtleties which distinguish it from the reaction diffusion. There are many similarities as well, but they're very crucial to the differences. For those who are not familiar, one of them is that the instability, like Turing, for example, here can be either convective instability, which will be swiped out at asymptotic times, or it can be an absolute instability. This is very important in a pattern selection mechanism. The other one is that we do need to pay a little bit more attention to boundary conditions. I mean, in reaction diffusion, we just assume periodic, or Neumann, or whatever. Here, we cannot do that. I mean, we can do that, but we need to be extra careful and understand how it really impacts when we go back to a realistic situation. Because flux and outflux are very important and will play a role, as I'll show you, in pattern selection process. Okay, so let's go back to our picture, and let's at first assume that we have an infinite system. So we look for uniform solution first, and then, as Alan Chapnis told you on Tuesday, we have a degeneracy, because it's a mass-conserving system. We cannot change the mass of motors. So we have a freedom of choosing one of them, and then choice of one of them will dictate the other two. And this is a very tricky issue when you don't have a very specific uniform state. So you do the analysis, and now in the analysis you play with one of them, and I chose MF as a free parameter. So the dispersion relation, as you see, it's stable until a critical value, and then it becomes unstable. And because it's a degenerate system, then you have also this zero mode, which means that you have degeneracy in uniform solutions. Now, in this type of bifurcation, the bifurcation can be either supercritical, so the new solutions will be stable, or it can be subcritical, and the periodic solutions will be unstable. They can then stabilize at some point, but at first they are unstable. What it means, it has a very important consequence, and you can test it. So you start with a small amplitude Q at the beginning, and what you get is a very large amplitude at the end. It means, and the amplitude is order of 1, which means that you're not really in the supercritical case, but you are in the subcritical case. So if you start with a periodic solution inside this hysteresis region, which has a very small amplitude, you will indeed decay to the uniform state. If you increase a little bit, then you will jump to here, and this is exactly what you see in this simulation. But again, without giving you the details, it is also known that in hysteresis region you can get isolated states, like the ones that Alan Chapman was referring, and these are excitable action potentials, if you wish. And if you initiate your system with only one perturbation, it will travel exactly as these waves. And this is also happens in this region. So in principle, we already now have all the machinery, and now we need to connect it, how cell knows how to distinguish between the two, how the selection really happens. We do have all the ingredients in the model, but now we need to connect it to the biology. So again, we have a multiplicity of states, we have our model, and we need to now specify the boundary conditions. So here, what we are trying to do is to keep fidelity as much as possible to the biological system. So we keep here the left boundary very low, and we take MF that it will have linear stability along all the domain. And here we do something different, because all these guys that travel to here, that they just cannot accumulate here forever. Otherwise, the philopodia will explode. So we just put some transition rates of conversion of these guys at the tip, either to freely diffusing, or to guys that as if they collected information and go back to the cell itself. So these are very simple options. It can be definitely more complicated, as I'll show you later on. More than that, we have another condition which also expressed here, is that if we can have conservation of flux of the motors. I won't spend too much here. And from the conservation of flux of the motors, we can have another removal of degeneracy. And it will play a significant role also here. I'm not going into details. If you have questions, ask me later on. So, two options of a selection mechanism. One, if we take the extreme of beta x equals 1 and 1 for 0, so this is the line of flux conservation, and you see here that we have traveling waves which are emitting from this uniform state, and here what happens is that we have pulses that are being generated here just as a pacemaker. Okay, so after this pulse propagates, there's another one that is being generated, and these are completely two different situations, and you see it also in the profile itself. How it really happens? The pacemaker is very easy and pretty generic, because again, from a mathematical point of view, it's related to homoclinic global bifurcations, and therefore really the model is not has to do with any details. I mean, you can get other ingredients, and then you'll get exactly the same behavior. Here, it's questionable, because you may think that it's a linear instability, but it's not. Well, it's also a global bifurcation, which starts from a heteroclinic connection, not homoclinic like the pulse, but heteroclinic between two uniform states. So at first you have a front, and then it becomes unstable, and it always becomes unstable in this regime because you can show that this region of flux is always unstable, and it starts to emit waves, and this is exactly the dynamics that leads you to this situation. Okay, so now if we play a little bit with beta to other values, we can go from traveling waves very smoothly to the situation of pulses. And again, this is really robust because it's based on global bifurcation. So it's very insensitive to parameters or to details of the model itself. You give me a different polynomial term or some saturation, I will now show you that it will eventually, I will find exactly the same condition. So if we now compare, for example, between beta is 0 and 5, so in a space-time plot, you see here the pulses, which are a little bit larger and more spaced between themselves, and you have the traveling waves here. And now, let's compare it to experiments. And experiments, in principle, you see almost the same behavior, where you have here these structures like here, and here propagation, which is more resembling to this situation. So in principle, we were quite happy, but it's not enough. I mean it's nice. It's a nice result. We published that, but I think there's much more to it. We did some, by the way, analysis. I don't have the time to go, but you can also compute the properties of asymptotic pulses. Again, these are just details that I don't want to spend too much time explaining. But what is important here is the outlook. And I really like the saying of Turing in this sense that if we can see that we need to do much more than we already know, then this is a real reward of mathematical biology. It's not when you claim that you solved the biological problem, but because I don't think that it's really possible. I don't think that mathematics will be ever that helpful to really solve cardiac fibrillation or some other medical issues. But it should open new philosophy and help little by little to promote the medical biological field. And if we look here about this behavior, then the obvious kind of conclusion will be that the boundary conditions are not just fluxes of conversion. You need to model them as their own entity, and they have to be dependent on time. They have to be dependent on specific chemical reactions. It's not just a simple problem. If we really want to understand what happens here, we gave kind of a recipe, but now we need to look into the details. And the details, the most important details, are at the tip. So if you want to really better understand the problem, you need to look at the reactions that happen at the tip. Okay? The other thing is a little bit more even tricky and interesting. In many cases, these philopodia, sometimes they grow and sometimes they don't. And this is exactly connected to the talk by C.R. Letaz yesterday. I'm sorry if I mispronounce it, which was very stimulating. And this is another example of how these philopodia, together with actin filaments and these motors, may impact this initiation of these structures. Now, we tried already to approach this problem by reaction diffusion elasticity problem, and what you see here, in principle, these are experiments, and here you have decay of the philopodia, so it's not a linear instability, it's a nonlinear instability, and here you do have initiation. Now, if you approach that with the respect of modeling a membrane plus reaction diffusion terms that control the polymerization of actin filaments from disordered to ordered situation, then you may end up with not really that problematic equations. Here you have reaction diffusion, and this is just a variation of Helfrich Hamiltonian, which is a standard Hamiltonian for membrane dynamics. And if you solve that, you really can understand, in a very qualitative way, of course, how really the mechanism of initiation works. But what's really missing here is these guys are highly dynamic. And this model cannot capture that. This model can explain only the threshold and the instability type. But if we want to incorporate the dynamics, we probably need to incorporate kinetics of molecular motors. Okay, so it really goes very nicely to different problems, and I think that also in cell polarization, when you have reaction diffusion properties coupled to the membrane and this different morphology, this is exactly the same approach can be applied here. The details, of course, may be different, but the approach and the most basic qualitative insights, I think, can be derived from here already. So at the end, I know I'm getting to the end. I definitely want to thank my collaborators, Nir Gov, who is from chemical physics in Weizmann Institute. Moshe Naoz was the student who wanted to ask me questions about the pattern formation, and we ended up with this project. And I'm really grateful for this smart guy that really introduced and pursued very difficult problems and initiated everything, in fact. And the experimentalists from NIH in the group of Bashara Kahar, and of course the experimentalists, it's always a very long list, so all these guys contributed. And if I want to sum up, then hopefully that I showed you to some extent that reaction diffusion advection approach is very appropriate to tackle problems of transport, and not, of course, also of myosins, but it's more general. You can really efficiently look at chemical reactions and other problems if you just configure a very simple set of equations. And you base all your understanding on global bifurcations and not on local bifurcations. That's very important. If you restrict yourself to linear instabilities, then it's model dependent. But if you go to global bifurcations, you need to pay less in the details of the model. The outlook, I'm sorry, again, the details is that we can distinguish between traveling waves and pulse trains, and this is being mostly by selection by the boundary conditions. In other systems, we can talk about other removal of degeneracy. Here, the appropriate one, as we believe at least, is through the boundary conditions. There are many aspects of removal of degeneracy, but the removal of the degeneracy can be very critical in any pattern formation in biology, as also Veronica and Alan expressed in their talk. The next thing, at least here, to my opinion, will be a rigorous modeling of the tip. It's not just a flux of conversion, it's time dependent. And we need to spend more time if we want to understand that. And I think that most of us do want to understand, because if we want to understand communication between cells, motility, many biologists say that relation to cancer is really something that we need to understand, the philopodia and the myosins, how they really work there. Again, I'm not a biologist, so I definitely cannot confirm or negate that, but it is something that probably need to be tackled. Transport of myosins and contractility, the philopodia are very dynamic objects. I treated them as static ones, and we verified that our results are in accord by just changing the philopodia size and looking at generic features, and that they do persist. But contractility is very important, and we do need to understand or put a little bit more effort how to make a theoretical framework that will capture that. And by saying that, we probably need to make more complicated models. More complicated models means much less elegant mathematics, unfortunately. So if you guys looking for elegant mathematics, forget it. I don't think that in biology you can get elegant mathematics. But it is rewarding on many other aspects. I think that that's all, folks. If there are students or that looking for positions or postdocs that are looking for challenges, then I'll be happy to talk to them. But besides that, I'll take questions. Thank you.
So in your model, you basically build in the jamming by having quadratic or polynomial interaction.
As simple as that.
Is there a chance to trace that back to connect the actual parameters to molecular processes?
I believe yes, but it will be very difficult because you need to kind of track concentrations and count molecules. I don't think that any biologist that value his work and his time will spend or devote that time to this project. I doubt it. I think it's too much effort, and at the end you get, let's say in the best case scenario, exactly the same conjecture of kind of polynomial type reaction. Who cares in biology? Biologists wouldn't care about that.
No, actually I have to, my experience is if you can relate what you do to directly to a molecular process, you are taken much more seriously and you can publish much further in biology.
Yes, but if you do it prior to publication of this work, but after this work was already published,
you're right.
And we tried to make experimentalists to look into that. They said that they had much better things to do.
So keep in time while Christian is setting it up. One last question.
It's a biological question. What's
Be careful! I'm not a biologist.
what's known about the cargo of this myosins in the filopodia?
You mean
What is transporting? It's not transporting.
Chemical molecules. It can be calcium. I don't exactly remember which one you transport what, but it can be any calcium or other chemicals that are needed for the cell.
And so it's also possible that these myosins are pushing the filopodia outwards.
Definitely, this is exactly, yeah. This is you're absolutely right. This is exactly the point. It can also be facilitated into motion of the filopodia. You're absolutely right. It's more complicated than that because they carry also kind of glue, and they show that you have a polymerization and adhesion to the surfaces because of the transport of different chemicals. It's much more complicated, and it's only now being kind of understood following this work. People in Finland who work on filopodia, they say exactly what you. It's not just necessarily chemicals, but it's also mechanical properties of adhesion of the filopodia. And if you knock out, and they see that, if you knock out some kind of transport properties, the filopodia don't grow because they cannot adhere to the surface. So it's very complicated. Beyond my knowledge, definitely. And I have only intuition and understanding of what I was told, but you're absolutely right. It's much more than just cargo. Well, when I say cargo, it can have many purposes. Exchange of information, but also kind of infrastructure by adhesion to the surfaces and so on.
Okay, the discussion is very inspiring and heated, but we have to go to the second talk, which will be very interesting, from Christian. Thank you very much, Arik.
I don't know how to